5 o'clock, time to get started. Alright, I just had to check that my pen actually works this time. I have about 50% luck with that one. Just keep finding things. It's a good one. I like that one. All right, we're going to talk about Chapter 17, Gene Expression. There's quite a bit of information since I think we started Chapter 17 back on Wednesday. So I'm not going to get a chance to get to everything. So if I don't cover something that you're wondering about, worried about, have questions about, obviously ask questions here in session is more than fine. But if we get to the end of the session and you go, wait, we didn't cover this, email them in, come up and talk to me afterwards, right? Because there's a lot of stuff to cover and I'm just not going to be able to do it in 50 minutes. So don't think that because I don't cover it in session it doesn't matter. If he talked about it in lecture, it can be on the test, right? Always remember that. All right, our biology fact for the day. Everybody go ahead and try it. You cannot breathe and swallow at the same time, right? You have what's called the epiglottis. It's this muscle here in your throat that closes and opens depending on what you're doing, right? If you're eating, you want food to go down your esophagus, right? And if you're breathing, or sorry, what did I say? Yeah, esophagus. And if you're eating, you want it to go down your esophagus. If you're breathing, you want it to go down your trachea. Otherwise, you get food in your lungs, right? Or air in your stomach. Air in your stomach doesn't sound that bad, but you can die from that. So this little muscle kind of flips down to cover to cover the trachea. So if you're eating, it flips down as you swallow, and then you don't get a piece of a hamburger down in your lungs, right? I think we can all see why that would be a problem. It doesn't necessarily keep air from going into your stomach, but there's different things that keep air from going into your stomach, which is also bad. Anyway, go ahead and try it. Try and swallow and breathe at the same time. Can't do it. The study tip for the day is to do 5-minute daily reviews, right? Especially with finals coming up. Okay, you're going to be tested on stuff from Chapter 1. How many of you remember what's in Chapter 1? Yeah. So finals are in what, slightly over 4 weeks, 5-ish weeks, right? If you wait until December 8th to start studying for your final on December 9th, you're going to be in big trouble. But if starting today, you go back and just spend 5 minutes reviewing as much as you can review in 5 minutes from Chapter 1, Chapter 2, Chapter 3, and so forth, by the time it comes crunch time for your final, you're not going to have to review 30-something chapters at once. Okay, you can just do a brief overview since you've been doing what's called spaced repetition. Okay, so start incorporating 5-minute daily reviews in. Get ready for finals now. Okay, before we start on this clicker question from class that I think only 40-something% got right. Does anybody have specific questions or material that they want to look at? Just go over initiation code. Yes, after this question. I will explain the answer for the over 46% of you, and 46% of you may have been guessing. I'll explain why that's actually the right answer. Anybody else? Is everybody just dying for this question? All right, so who remembers first off what the answer was? It was C, right? Within the mRNA. The wording, I think, is one of the trickier parts of this. It basically just means not on either end, right? It's just anywhere between the 3' and 5' end. Right? The promoter is its own separate thing, so we already know. Not that. What's at the 5' end? Does anybody know? It's a cap. So if there's a cap there, what's not there? An initiation codon, right? What's on the 3' end? The poly A tail. If there's a bunch of A's there, what's not there? The initiation codon. And then within the first intron, why not in the first intron? We're starting, initiating, why not in the first one?
That's only part of it? Only has part of the codon in it?
It has codons in it, but it doesn't have the initiation codon. Why not?
Because introns get cut out.
Yeah, it's a little counterintuitive. Introns leave. Introns get cut out. And exons, you think, sounds kind of like exit, so it should leave. It doesn't. Exons stay in, right? It's a weird, I think it stands for like interleaving group, and I can't even remember what intron and exon, there's like a full title that makes sense once you know them, so I should have known them. But exons stay, introns leave. So if we remove an intron, why would we not want to have a start codon there, an initiation codon, sorry? Yeah, if you cut out the initiation codon, it's like not putting gas in the car, right? Everything else can work just fine, but you can't start the car. Or like not having keys, right? That's step one. So do you guys at least see why A, B, D, and E are wrong? Okay. So we have our original DNA template, right? We cut out our introns, we break it down just to our exons. We have our untranslated region down here, and then somewhere down here in the 5' end would be also that cap, which is also untranslated. The caps just serve to keep the mRNA from getting broken down, right? It's like the aglets on your shoelaces. If you just have a shoelace out there, it's going to start falling apart. So mRNA kind of works the same way. If you just have naked mRNA out in the cell, it starts to fall apart. So you put these caps on it, right? So our initiation codons are just kind of peppered throughout the mRNA, depending on which gene you're looking at and how many genes you've got. So they're just spread all throughout the RNA.
So is there only one initiation codon?
Per gene.
But is there only one pattern? Is it only AUG?
AUG is the only one that codes for a start, yeah.
Okay.
Yeah. That way you don't accidentally start multiple times or in the wrong spot, yeah. So that one only gets one. I think that's the only one. No. There's one other amino acid that there's only one sequence that makes it, but that doesn't matter. It's pretty much the only one that AUG is the start codon. Okay?
There are different stop codons though, right?
Yeah, I want to say 3,
Yeah, that's
right?
how many there were.
3? Okay. So there's 3 stop codons. I don't know, just because.
Oh, I was just going to ask that. Why are they 3?
Just because. I don't know. You don't need to know. Probably just makes it easier to stop. I don't know. Other questions? That makes me sound dumb. I don't know. It's not going to be on the test. So, all right. So does that answer the clicker question? I saw less than 50% get that right, and I thought we should probably go over that. All right. Questions? Okay. So we are going to talk about gene expression, Chapter 17. That's all this is about, right? 3 major portions. I basically have time to talk about one of them since it's so large. Translation, in my mind, is the more complex, more difficult, more conceptual process. So we're going to talk about translation today. That does not mean that you don't need to understand transcription and processing. It just means that's what I chose to focus on for the session. And if we have time at the end, then we'll look at some transcription. Cool beans? All right. So the whole purpose of translation is to decode information, right? When you guys think of translate, what do you think of? language, right? You like English to Spanish, Spanish to German, German to Japanese, right? We take one language, we take the information in that language, right? And then we translate it so that it makes sense in a different language, right? So I guess we should go back to just briefly say, transcription is the act of taking an idea and writing it down, okay? In DNA, it's the act of taking genetic information and writing it down in the form of RNA, right? That can actually be read, right? It's like thoughts to words, right? I can have thoughts in my head and that's all great. But if I want other people to have them as well, I have to say them or write them down. So mRNA is the equivalent of speaking or writing your DNA out, right? So we transcribe it into RNA. But does RNA automatically equal, like if I have this strand of RNA, does that automatically just mean I have brown hair? No, what has to happen first? We have to translate it, right? So it's like if I'm just sitting here and talking, okay, does everyone in the entire world understand what I'm saying? No, not everybody speaks English, contrary to American beliefs. There are multiple languages out there, just so you know. You have to translate it, right? So we have to take our RNA and translate it into useful code that becomes amino acids, right? And then we chain all those together and we get our proteins. So we take our mRNA, we translate it so that we can have our tRNA form a polypeptide chain. What's a polypeptide chain? It's made of amino acids. What's a whole polypeptide chain, though? It's a protein, right? Why do we need proteins? Everything. Eye color, hair color, blood cells, skin cells, brain cell, everything is made of proteins, right? So this is essential to life. All right. So who does the heavy lifting? Is the tRNA. Does anybody remember what tRNA stands for? Transfer RNA, right? Not translation RNA. That's kind of a common misconception because we think, well, it's in translation, tRNA, it makes sense. It's transfer, I almost said transcription. Transfer RNA, okay. So it's what links our codons to our amino acids, right? Where are the codons? In the mRNA, right? Where are the amino acids? Not yet. Before they're on the tRNA, it's in your cytoplasm, right? You just have amino acids floating around, okay? We'll look at how we get the right one in the right place in a minute here. So this is the middleman. He takes the mRNA and he says, okay, this is what I want to read. He takes the tRNA and says, this is what I want to attach, and he works in the middle to facilitate the whole process. So let's look at the two most important parts of it, right? There's a lot going on. There's plenty of things you could look at. For this class, we really just care about this amino acid attachment site right there on the 3' end and this 3-letter anticodon on the bottom. When you think anticodon, what do you think of? Codon, but more specifically complementary or like opposite to codon, right? So just the reverse. So what codon would that anticodon pair to? I heard TTC. Almost. UUC. Remember in RNA, it's U's instead of T's. Everything else is the same, A's, G's, and C's. But it's U's instead of T's, okay? You may see a test question where, for example, UUC and TTC are an option. That's when you stop and go, okay, are we dealing with DNA or RNA? Make sure. If you're dealing with RNA, then you know it's U's. If you're dealing with DNA, then it's T's, right? So just make sure you know which one you're dealing with. Questions up to there? Okay, this is not what tRNA actually looks like, right? This is if we like flattened it out and then colored it and put letters on it and stuff, right? This isn't what it actually looks like. And neither of these are really what it actually looks like either, but this one on the left is, you know, sort of a 3D model to give you an idea, kind of what they look like. And then the textbook uses this like cute, almost like cartoonish one. It is nice because it makes the two most important features prominent, right? Our amino acid attachment site that kind of just looks like a single hair up there, and then that anticodon on the bottom, right? So just know what they are and what they do. So what's an anticodon? It attaches to a complementary codon. So it's just attaches to the codon that has the inverse of its 3-letter sequence, right? What's the amino acid attachment site, if you had to take a wild guess? It's where an amino acid attaches, right? How does it attach? I've given you a hint here. Nobody wants to say it because they can't pronounce it. Aminoacyl tRNA synthetase, right? It's called aminoacylation, or the easier term is charging, right? So how it works, we have this tRNA synthetase. In this case, it's the tyrosyl tRNA synthetase, meaning it works with tyrosine, that specific amino acid, but it could be any, right? Now, it has a very specific shape. It's quite handy that it's, you know, cartoonish for our case. They obviously don't look exactly like that. But notice how the specific amino acid fits into that specific spot, right? A square amino acid or a triangle amino acid or a circle amino acid wouldn't fit, right? And then it doesn't really show it very well here in this picture, but it's got to have the right sequence down there in the bottom to match up with the tRNA. Why?
So that in the end, it can make the right glycoprotein.
Yeah, if we just stuck any tRNA, so that's a tyrosine tRNA. If we stuck a leucine tRNA in there with a tyrosine, then we're accidentally going to put a tyrosine where we wanted a leucine, and pretty quickly we're going to develop every problem in the book, right? So you have to get the right amino acid on the right tRNA, right? It doesn't just matter that you get the right tRNA in there, right? What if it's not got the right amino acid on it? Okay, so they both have to be right. So essentially, our tRNA fits in like a lock and key, and that amino acid fits in like a lock and key. We use some ATP, right, to, I guess, kind of like fuse this amino acid onto the binding site, and we make our full-blown tRNA with an amino acid. So what's it called? What do we call a tRNA that has an amino acid on it? Charged. It's a charged tRNA. Okay, if it has an amino acid attached to it. You can kind of think of it as if ATP is like electricity, we put ATP into it, we've just charged the system, right? We've just put energy into it. Okay? Questions up to there? It is freaking cold. Am I the only one that's cold? I'm wearing 2 jackets. All right. This is, as Dr. Pogorski puts it, biology's Rosetta Stone, right? What's the Rosetta Stone used for? Translation, right? It had like Egyptian and Hebrew and Latin or something on it, and that's how they were able to make the link between those ancient languages, if anybody doesn't know what the Rosetta Stone is. So what he's basically saying is this, what do you want, a table here is how we know which amino acid we're getting out of which codon sequence, okay? So you have to memorize all 64. All right, just the amino acids. 2 people thought it was funny. No, that would be a nightmare, because there's 64 of them, right? You don't have to memorize the codons. You don't have to memorize even the amino acids. If you were to get a test question that was like, what amino acid would you get from UUC, right? You would be provided a table. I don't remember any questions like that, right? Where it specifically just said, here's the codon, what amino acid would you get? It's mostly because Dr. Pogorski knows that doesn't really test anything except your ability to look at a table, right? He tests more. If we took UUC and substituted, deleted a U and put in an A, what would happen? And you wouldn't have to know, well, it would turn from leucine to tyrosine or anything like that. Maybe you would have to know what kind of mutation that is or what kind of disease that might cause, right? And I'll give some examples of that.
So you say we don't need to know what the amino acids are, but we should know kind of the PRO, LU, THR, stuff like that. Do we need to memorize those, or is it just more direct?
No, just if you heard leucine, tyrosine, or something, glycine, valine, I don't even know all of them. There's like thornine and serine. I think that's arginine. If you hear like anine, just know it's an amino acid. So be familiar enough that you're not like, wait, what are we talking about? But you don't have to be able to what is ARG? And be like, oh, it's arginine. Nothing like that.
You did say that we need to remember that AUG is the start codon.
Yeah, I would be familiar with the start codon and maybe with the stop codons, just because they're unique. And before you ugh and groan, and this is the worst, it's 4 3 letters. It's 12 letters. So I don't really feel bad telling you that you should be memorizing 12 letters. It's not that hard. Okay? Now, after I just told you that you're not going to need to know how to do that, I'm going to make you do it anyway, just in case. Because his tests do change. So I would feel bad if I was like, yeah, you're not going to need to know how to do this. And then in the test, it's like, so what's UUC? And you're like, frick, he told us not to. So here's your codon table. I will help you with the first one in case you're like, I legit don't know what's going on in this table. So G, G, A. You go G, so we know it's somewhere in here, right? Then you take your second codon, not codon, base, and you know, well, I shouldn't have done that, huh? So now you know it's got to be in this box, and then we know our last one is an A, so it's glycine right there, right? You guys see what I did? Okay, take 20, 30 seconds, figure out the other 5, maybe share with a neighbor to make sure you actually did it right. Again, I know it's not rocket science. I know it may feel a little juvenile. I just didn't want the test to be the first time you saw a question like this. And I'm more than likely you probably won't even, but just in case, right? And it's not the hardest thing in the world. This may be all the practice you need on how to read a codon table, right? Just go through the left there, U, C, A, or G. Then go through the top, U, C, A, or G. Line those 2 up. And then out of that box of 4, pick U, C, A, or G. And there you go. All right. What do we get from UCA? Serine. What do we get from ACA? Threonine, or threonine. I don't even know how you say that one. Right? A, C, A. Threonine, something like that. What about CGG? Arginine. UAG is our world famous, where am I? U, A, G, stop codon, one of the three, right? What about AUG? It's methionine or the start codon, right? They're the same thing. So some tables will say methionine or start, some will just say methionine, some will just say start. So if you see just MET, mentally in your head say to yourself, that's also the start codon, right? So if you get a table and you're like, there's no start codon, why does it not say start anywhere? It's methionine. Okay? Questions there? All right, ribosomes are super, super fascinating, right? It's a macromolecular machine. First of all, what does macromolecular mean? Yeah, it's compared to other things in your body, it's actually pretty big. Ribosomes are pretty large. I don't know how big. They're obviously not like the size of a quarter or anything, but on a biological scale, pretty big, right? It's because they do a lot of work, okay? They allow for protein synthesis, okay? Yeah, that's all I want to get out of that. This is what I really want to look at, is the structure of a ribosome, okay? If you don't understand the structure of a ribosome, you cannot understand translation, okay? Because the ribosome does the translation, right? The ribosome is the guy who actually takes the Rosetta Stone and says, okay, eagle over mountain means happiness. I don't know how Egyptian works. You have 3 sites, the EPA, right, Environmental Protection Agency, but they actually go the other way, so it's APE, right? A stands for aminoacyl tRNA binding site, P stands for peptidyl tRNA binding site, and E stands for exit site. I don't like most of those names. I don't like 2 out of 3 of those names. So I start this with the disclaimer that these are not the real names. I call them the access site, the pass site, and the exit site. Because it enters, it accesses in that A site, it gets passed to the left into the P site, the pass site, the peptidyl, and then it exits in the E site, right? So again, real names, aminoacyl tRNA binding site, peptidyl tRNA binding site. I like access and pass. It's my mnemonic to remember it better, right? Your mRNA binds right there, right? Because we have to hold it down somewhere, right? We want to be able to stretch it out and read it. So it's like when you open a book, you know, you keep your fingers on the sides so that the pages don't close on you. The mRNA binding site is like the thumbs on the pages, right? It's just what keeps it open so you can read it. So we stick an mRNA right in there, and it's got all of its codons, right? A tRNA comes into the A site, the access site, the aminoacyl tRNA binding site. Its anticodon has to match up with the codon that's already there, right? Otherwise it won't attach, because we would put the wrong protein in the wrong spot. Okay, so if it's a right codon-anticodon pair, it attaches, okay? So you can call it the attachment site too, I guess. Attach, access, whatever. Just know that those aren't the real names. Then it passes over to the left, right? Well, I think technically first the amino acid attaches to that further growing chain. No, I did it backwards. See, even I get it backwards. The tRNA here gives the chain to this tRNA. So it kind of like shifts to the right onto that tRNA. And then this whole thing that now includes that, I know I'm making a nightmare of this, also passes over to the right. So let me do that again in case that was like an absolute nightmare. This tRNA has built a polypeptide chain, right? It detaches here and attaches to the new amino acid that just came in. So now our chain is here, but then we pass the whole thing to the right one, and so now we're back to where we started, but we're one protein longer, right? And then this one that was originally here, see how that amino acid's gone now? It passes over to the exit site and leaves. Okay? So tRNA comes in, matches anticodon with codon. Amino acids get passed back onto the new one. That one slides over, and then, it just passes back, passes back, passes back, passes back. And after it's passed back, it leaves, right? The tRNA leaves, and the amino acid chain grows and grows and grows. Then we hit a stop codon, and it just stops, right? And that amino acid chain that's now a protein is delivered to wherever it needs to go to perform whatever function it has. Did I say a hand?
What does the mRNA binding site do?
It's just holding the mRNA in place and kind of sliding it along so that we go 3 codons at a time.
Is there a purpose for the exit site, or like why does it move to the exit site and then leave? Why doesn't it just leave from the P site?
I think the easiest way to look at it is as the amino acid chain gets passed back and then comes over, right? If it left from that same site, it would take the whole chain with it. So it has to detach and slide over to kind of get out of the way, because it doesn't happen 2 tRNAs at a time. You're sending in thousands of tRNAs. So just having that extra exit site keeps traffic low, I guess.
So nothing happens at the exit site.
No, it passes over and pops out.
So when you move to a different codon, is it the ribosome that's moving over, or is it the mRNA that's sliding through the ribosome?
I'm not positive. I'm pretty sure. Which one does move? I don't know. Does anybody know?
He said it was the RNA that was moving through the ribosome.
So the ribosome's kind of stationary in the mRNA. That's what I thought, but I was I don't know, maybe the ribosome crawls along it. So the mRNA snakes through, kind of almost gets pulled through, I guess you could think of it. And the ribosome just stays there and reads as it comes by. Which would make it work faster if you think about it. Other questions? All right. So, to give you a better visual of what I just tried to draw in one picture, we have 4 pictures. So we start here. We already have a tRNA in place, and it's already got 4 amino acids on it, right? A new tRNA comes in, matches anticodon to codon. This chain gets passed over to the right one to now form the chain on that tRNA that just came in. This tRNA slides over to here and then leaves, and this tRNA slides over to here and is now in the P site, and we do it all again until we get however long a chain we need. It could be 5 proteins, it could be 500, amino acids, it could be 500 amino acids. I don't know how many amino acids are even in your standard protein, to be honest with you. Probably more than 4 though, if I had to guess. Questions? Is this just making sense? Because no one's saying anything. All right. I forgot to pass out the roll too, so let's do that. 8:30 on the front, 9:30 on the back. Yeah, if you don't ask questions, I'm just going to assume that you understand this. Yeah.
So is one cycle just with one tRNA, or is that with the whole?
Say it one more time. I'm not sure I understood.
So it says the one cycle.
Yeah.
So is that just talking about one tRNA coming into the ribosome and exiting, and that's one cycle, or is it talking about the entire mRNA going through the ribosome?
One cycle is for every essentially every new tRNA that brings in a new amino acid. So this is the tRNA that we started with. So it hasn't quite left technically, but one cycle is basically for every time a new amino acid gets introduced. So then the next tRNA to bring in another amino acid starts like a new cycle, I guess. There's not, I don't know, nobody's sitting there going, that's one cycle, that's two cycles. But basically, yeah, that's where it starts and stops. Other questions? On to mutations then, right? These are permanent, heritable changes in DNA. So you guys remember the clicker question from class about whether or not, I think it was UV radiation would cause damage to your DNA. Is that a mutation? What was the answer? No, not necessarily, right? What was the key word that was missing there? Permanent, right? If it had been a permanent, unfixable change in your DNA, that's a mutation, right? If not, we would mutate every second, right? There's radiation in this room right now, right? There's radiation in bananas. I found out the other day, and I can't remember the number now, you'd have to eat like 5 million bananas in order to die from radiation. But you would die at like 2 million bananas from like potassium overdose or something. Anyway, in case you're wondering how many bananas you could eat before you die. And that's at once, right? I don't know, like in your lifetime, I don't know how many bananas the average human eats. I've probably had 12. Like, I don't know. How many bananas have you eaten this lifetime? That makes it sound like we live multiple. We might, I don't know. I'm getting way off topic. Mutations have to be permanent, right? We fix mutations, we fix issues in our DNA all the time, right? We get radiation every time we go out into the sun. You get radiation when you go to a tanning salon. You get more radiation than you should when you go to a tanning salon, and your chances for skin cancer are like astronomical, so please don't go to a tanning salon. You might look good in January, but 16 years from now you might not when you don't have a nose. That's coming from someone who got skin cancer, so just try me. All right, to understand the effect of mutation, you must understand how genes are expressed and how their products create phenotype. What's that trying to say? That's a lot of big words. What's Dr. Pogorski really trying to tell us?
Is that you need to understand how a gene normally works. Easier to see its mutation.
Yeah. So if you don't understand gene expression, then how are you going to go ahead and say, oh yeah, well if that codon gets removed, if that codon gets added, if that codon shifts to the left, if this codon gets inverted, whatever, you're not going to be able to go, well then this is what's going to happen, right? You have to understand how genes are expressed before you can understand how they get expressed wrong. Okay? So this is a picture of a fly with a mutation called antennapedia. It has legs instead of antenna, right? It might just kind of look like weird antenna in this picture because you've never seen a fly that close up. Those are actually its legs, right? It has legs coming out of its face. It'd be like the equivalent of humans having toes where their ears are, right? This is pretty bizarre stuff, right? But this is relatively common in flies. Far less common in humans. I don't know if there's even been a human to have toes for ears. Somebody Google it. This shows you how old Dr. Pogorski is. He's had the same slides since 2007. It's always teenage. There's been like 3 other Teenage Mutant Ninja Turtle movies. X-Men, Deadpool, like all of these are mutations. He picks the 2007 cartoon. I haven't even seen that movie. Probably got like 4% on Rotten Tomatoes. We should have done a clicker poll. How many of you have seen the 2007 Teenage Mutant Ninja Turtles? All right. This is what I really wanted to look at. Mutations that alter one or a few nucleotide pairs are called point mutations, right? Because they happen at a pretty specific point, right? It's not like an organism-wide mutation. It happens usually on like one or two nucleotides, right? That's what we focus on in this class, okay, is point mutations. Be familiar with that vocabulary word. One type of point mutation is base pair substitution, okay? There's 3 kinds of base pair substitution, okay? Silent, missense, nonsense, okay? So this one is a silent mutation. Who can tell me why?
It doesn't change the phenotype.
Okay, perfect. So we have an A here, right? Or sorry, we should have had a G in our original DNA, but something happened to our DNA. We ended up with an A instead of a G, which means we ended up coding for a U instead of a C in our RNA, right? And so GGU coded for glycine. But it just so happens that GGC also codes for glycine, right? Remember the degenerate nature, the redundancy of the genetic code. More than one sequence codes for the same amino acid. So we got lucky this time. GGC and GGU both happen to code for glycine, so we still get the same protein and nothing happened, right? There's probably a decent percentage of people in this room who have silent mutations, right? Something changed in your genes, but nobody knows because nothing happened, right? Doesn't mean it didn't do, it doesn't mean that the mutation didn't happen. It just means no phenotype change happened. Then we have a missense mutation, right? We should have had, I'm trying to see where we are. We should have had a C in our original DNA, but we ended up with a T, right? Which means we ended up with an A instead of a G, right? So in this case, GGU coded for glycine, but GGC codes for glycine, AGC codes for serine, and we actually get a different amino acid in our chain. We're going to get a different protein, right? It could be something as harmless as a change in your eye color, right? Or it could be as harmful as sickle cell anemia, right? Is everybody following me so far? The worst one is the nonsense mutation, right? We should have had a T, but instead we had an A, which means instead of making AAG, we made UAG, the dreaded stop codon, which means we only made methionine, right? Which is just the start codon. So methionine on its own, not going to do anything. So if this were, your gene for heart cells, now you have a problem. Because you don't have any proteins, you would not grow a heart. That's a somewhat dramatized Gray's Anatomy example. But you wouldn't form whatever protein that you were making. And so maybe you wouldn't be missing an entire heart. You could have a hole in your heart, probably. I don't know. I'm not a geneticist. I'm just making up examples.
So with the nonsense, is nonsense just when you have the start codon and then on the next one is just the stop?
No, you could have 4 or 5 amino acids. It's just when a mutation causes a stop codon when it shouldn't have. It could be even at just one amino acid too early. It's just a stop codon before it was supposed to happen. Does that make sense?
So could one of the nonsense mutations be a start codon instead of a stop codon? Like a few of the codons later, it's a start codon.
So you're saying like we've put down like 2 or 3 amino acids and then we get another start codon?
Yeah.
I've actually never heard of that. I don't see why it couldn't happen, but I also could not tell you what it would do. I don't know if it would like start another protein. I don't know. It's a good question. I doubt you would be tested on that, but that's a valid point. I don't know what would happen. You would get twice as much protein. You'd be huge. I don't know. Other questions? I thought I saw another hand. Was I distracting? Yeah, that's fine. At least it's entertaining, right? Insertion and deletion mutations. Also point mutations, okay? The difference here is we're not substituting, right? We're not changing an A into a T or a G into a C or anything like that, right? We're actually physically removing a codon or adding an extra codon into the sequence. These usually end up being the worst of all, right? And you'll see why in a second. So here's our wild type DNA. I believe this is, an insertion. We take an extra A and we stick it in here, right? So it used to read TACTT. Now it reads TACATT. Do you guys see what happened? We stuck in that extra A, right? This coded for UAA. So in this specific case, it's a frameshift causing nonsense. But it is an insertion frameshift that caused a nonsense mutation. And insertions don't cause just nonsense, right? They can cause missense. They can even be silent, though that's pretty rare, right? Because Dr. Pogorski showed in class if you have the cat ate the rat, and we stick in, let's say, a W right there. Now we have I'm going to see if I can even do this right. Right? That makes absolutely no sense now, right? You've completely destroyed. If you did that to a book, right? If you just changed like if you just stuck another letter into the second word in like War and Peace, right? But that meant that every word had to get shifted over one letter. The entire book would be insanity, right? It already is, but it would be a nightmare. You would not be able to read it, right? So insertions they just throw everything off so hard, right? Deletion's the same way. We cut out that A. So it used to read, we'll start here, TCAAC. Now it used to read TCAAAC. Now it reads just TCAAC. So we're missing an A, which means we end up missing that U. And when we would have coded, I think that's phenine or theanine, I don't know, we end up coding for leucine, right? So how is this different from a missense mutation?
It affects the whole polypeptide chain.
Then every other amino acid following it is also going to get changed. So it's the same as if we had, the cat ate the rat and we cut out the E. Now we have, am I even going to be able to do this? Right? Still makes no sense. And each following amino acid is going to be screwed up as well. And you're going to get a protein that is massively different than what it was supposed to be. So for example, in a fly, right? Its antenna gene probably had an insertion or deletion mutation that caused a complete change in protein, right? And so instead of growing antenna out of its face, it grew legs. Do flies have faces? Head out of its right here. It grew legs, right? It had the wrong protein, and it coded for legs where it should have coded for antenna. Does everybody see how these are working? So base pair substitution, right? Silent missense nonsense, right? No what each of those three are and know how they happen, right? And then insertion and deletion is what it sounds like, but just be able to identify it, right? So if you were given just that wild type and then that, I guess I should not point at the, if you were just given that, I guess maybe just this whole thing. We'll just say the whole thing. If you were given just that, be able to identify whether that's an insertion and deletion and what it would do, right? Would it shift everything to the left? Would it shift everything to the right? What's it going to do to our sequence here, okay? Does that make sense? Is everybody following what I'm saying here? All right. That's it for today. If you have questions, email me, come up and talk to me. Ask Hillary tomorrow. Ask Chase tonight. Where'd that roll end up? Did everybody get the roll? Raise your hand if you did not get the roll. Excellent.